Thanks to IBA for sponsoring this episode.
Hello, welcome back to the newest edition of the JNM podcast. My name is Ken Herrmann, and I'm super excited to have three outstanding contributors, actually from three different continents. And to start with our colleague from the West Coast first, it's my pleasure to introduce you to Professor Brenda Sandmaier. She is Professor and Deputy Director of the Translational Science and Therapeutics Division at the Ferid Haddad Cancer Center, and she also serves as Professor of Medicine in the Division of Hematology and Oncology at the University of Washington, and she conducts quite a few clinical studies involving 211 Astatine. Welcome, Brenda.
Welcome. Thank you for inviting me.
The next participant is Dr. Ferid Haddad. He's a Managing Director of the JIB Aromax, and he's the one probably who has the most experience in Astatine in this call. He's a researcher in nuclear physics. Welcome, Dr. Haddad.
Thank you very much. Welcome, and thank you very much for inviting me.
And last but not least, the person who stayed up the longest today, Tadashi Watabe, is Associate Professor and Lecturer in the Department of Radiology at Osaka University. He serves as a Principal Investigator for a couple of physician-initiated clinical trials using Astatine 211. Welcome, Tadashi.
Thank you very much, Ken.
So the reason we chose the topic Astatine 211 is that there has been a recent article published in the Journal of Nuclear Medicine by Dr. Richard Zimmerman with the title, "Is Astatine 211 Really Happening?" And to kick it off, I want to ask all three of us with a very quick statement asking you where you perceive 211 Astatine in 5 years from now, which means pretty much in 2030. And maybe we start first with Dr. Haddad, because you're the one who knows the most regarding the likelihood how successful the production route will be.
So in 5 years from now, I think that Astatine will be really available. I mean, there are several cyclotron providers that are making specific machine for that. So there will be lots of Astatine all over the world. And I expect that some clinical trials that are already starting will give very good results. So it will be really an interesting isotope to look for on alpha therapy.
The nuclear medicine perspective next, Tadashi.
Thank you. So last week I attended SNI meeting in New Orleans, and I presented about the results of sodium oxide trial for radioiodine refractory thyroid cancer, and I got a great response from the participants, including industry people. And so far, the clinical efficacy data about astatine is limited, but from these 5 years, we will have more clinical efficacy results, and of course, we will have more production sites and supply. So it will be very promising 5 years from now.
And what is the clinical perspective, Professor Sandmaier?
Well, the clinical perspective, I feel like, at least for the work we're doing, we're limited by the production. And it makes me very excited to know that you experts are working on the way to bring this so that other places can have access to astatine. We've done clinical trials since 2017 with our first in human patients started in 2017 and treated over 50, actually close to 60 patients with both malignant and non-malignant hematopoietic disorders as part of conditioning for transplant. And like I said, I feel like we're only limited by the availability, though I see by reading and talking to people that the production problem is something that is surmountable. Because that's the question I always get. Well, it's good you can do it. What about, other places? Can't we do your studies?
This is a perfect trajectory to one of the first questions where we want to dig deeper. And there's nobody better who can answer than Dr. Haddad. So can you update us a little bit, first of all, what are the different production routes? Astatine has been around for quite some time, but when you follow the news, it makes me believe that now is the time that becomes really widely or more widely available?
Yes, so the Astatine-211 is produced using an alpha beam that interact with the bismuth target. So bismuth is very abundant, so it is not a problem. What is more problematic is the availability of alpha beam, because most of the machines that are used for medical isotope are made of accelerating proton. So there have been in the past, long time ago, there have been many machines making multi-particles available, multi-particle projectile available. There have been some that are being constructed at the moment, but the real step forward is dedicated machines that are being developed for Astatine-211 production. And there is at the moment all the cyclotron providers that are looking for this kind of machine that are specialized to Astatine-211 production. So that will make this astatine more available, and that will also reduce the cost as it will be optimized for Astatine-211 production.
Can you provide us some details? Just because, many people who listen to us obviously are just regular nuclear medicine physicians like me. So what MeV number is necessary, just to have an idea? Probably the 18 MeV I have for my fluor 18 production is probably not sufficient, right?
Yes, what you need is 28 MeV. So first you have to add the right energy, which is 28 MeV. You have to have the right particle, which is an alpha particles. And then you have to make sure that everything goes quickly, because from the production to the use, because the half-life of Astatine-211 is only 7.2 hours. So you also need to automatize things and to make it as an industrial production. So this is why having this new development, both in the cyclotron, but also on the chemistry for the extraction purification, and also in the radiolabeling, all these things will help to have the radiopharmaceutical quickly after production and enlarge the use of this Astatine-211 radiopharmaceuticals.
But if I understand correctly, this also means in case that you really bring one of these drugs to market, you really need a dedicated network of cyclotrons who are 100% dedicated to Astatine production. Is this correct? Or?
Yeah, that's true. That if you really want to make it available, you will need a network. For example, in our case, we are producing and shipping in France, but also in Belgium and Switzerland and in Portugal. So there is a limited area where we can provide our products. So you will need several cyclotron or accelerator to cover a whole continent.
Can I ask a question? Is there any regulatory issues with the shipping? Are they done by flights, or is it done by ground transport? Because
no, the point is that you have to be quick. So if you use a plane, then you have to make sure that you don't lose time when you board the Astatine on the plane, and then you get it after. And if you cross borders, then there may be some also issues. So this is more an optimization all this time that will be very important. But coming back to the half-life, very important thing with half-life is the fact that because you have a limited area where you can provide your product, that means that people and researchers are really working together, because what you are doing in your area will not have an impact on another area. So there is a world Astatine community that have been set up where researchers from all over the world work together in order to push the field to the radiopharmaceutical.
So I read that around 10 hours of transportation, no matter if it's train, car, or plane, but this is realistic. It should not be more, otherwise it becomes quite tedious. Tadashi, nuclear medicine, for many years, dealt almost only with iodine-131, a little bit of yttrium-90. Recently, lutetium has come around. Now we talk about lead and actinium and terbium. Now explain me, why do we need Astatine, and why do you believe so much in Astatine? You're really building your career on Astatine. That's why I would like to hear what's the point.
Yeah. Thank you very much. So the why I started using Astatine is the reason is simple, because in Osaka University we have a research center for nuclear physics, and they can produce Astatine with their cyclotron, and So the availability for the astatine is very rich in Osaka and for Japan. But from the point of the nuclear medicine utility, I think the short half-life of 7.2 hours, it's challenging, but I think it's also advantage because we can deliver the high radioactivity to the target in a short time. Because a longer half-life nuclide needs the reduced radioactivity for the injection, so we can get the response, very quick response after injection. That's, I think, the advantage of using astatine.
What about the chemistry? Is it correct
Chemistry.
that this chemistry is very similar to the iodine chemistry, which makes it we don't need chelators anymore? Is this correct, or?
So we don't need a chelator. We can directly label astatine to carbon to benzene ring by some covalent binding. So for example, we can label astatine to amino acids directly. So that's the difference to the chelator binding compounds.
Thank you, Tadashi. Professor Sandmaier, you work at the University of Washington, and I think they have a quite good tradition in yttrium-90 radioimmunotherapy. If I remember correctly, also the IOMAP, the iodine-131 in malignant hematological diseases was used a lot there. So now you come up with the next, let's say, interesting radionuclide. Tell us, what motivates you to work with astatine to another level as an oncologist?
Well, yes, we do have the long history with I-131, oh gosh, from the late 1980s. I mean, I've been here since 86. And so we've been involved in those clinical trials, well, preclinical work, clinical trials. And we also did work with yttrium-90, which is a pure beta, which has its advantages. But the problem is the path length is long enough, so you do get scatter off of your target, because we use antibodies to target the isotope to where we're going. And it's primarily used in hematopoietic malignancies. So if you think you want to have something that's like a laser and targets the hematopoietic cells, not surrounding tissue. And I think astatine, an alpha-emitter is perfect. Preclinically, we start with bismuth, but a 46-minute half-life showed proof of concept, in our preclinical models, but we knew that was not going to be a practical isotope, and that's when, with Scott Wilbur doing all the chemistry, we started with astatine. And to me, it's a perfect half-life, because with 7.2 hours, you can ablate the hematopoietic cells, and we have to put in new stem cells, hematopoietic cells, to rescue the patient as part of transplant. And if you have a long-lived isotope, it's not ideal. And you really want the isotope in and back out. And 6 half-lives is basically just a couple of days. And the other advantage to us is you didn't have to shield the same patient as we did with I-131. We had lead-lined rooms. And, you expose the other healthcare workers along with family members if they're able to go home. But I think with astatine, a lot of those problems are solved.
So which antibody do you use? You mentioned up to 60 patients you have already treated, all prior to stem cell transplantation, or?
Yeah. We have used an antibody that targets CD45, BC8. It's the one that we've done with the I-131 studies, the legacy studies, yttrium-90, and it's the one that they did in the Sierra trial with Actinium Pharmaceuticals. They licensed the antibody, and that was the antibody used in the trials, and so that's what we're doing. But that's a murine antibody, and a lot of our work right now is to make a human or humanized antibody.
But thanks for highlighting this, because the idea of really eradicating the bone marrow niche prior to stem cell transplantation is a beautiful one, right? But the problem of the iodine-131 with almost 8 days of half-life is that, first of all, the patients have to stick around for very long, right? And the second thing is they're still quite radioactive when you actually want to transfer them to the transplant department?
Well, it's not just a half-life. I want to clarify. It's not the half-life of the isotope per se. It's the biologic half-life because, they excrete it, it targets. And so the biologic half-life is actually shorter, but still it's much longer than astatine than when you want to clear it. And because total body irradiation was always a part initially and hematopoietic cell transplant, but now there's a lot of movement to chemotherapy. But high-dose chemotherapy, again, has toxicities to other organs. So if you could just target the hematopoietic cells, that would be great.
Super. Dr. Haddad, I want to ask you nevertheless two complicated questions. First of all, I read about the opportunity of having generators, radon, astatine generators. Is something real or is this science fiction? And the second question is, if I understand correctly, and one of the decay pathways, there is also a radionuclide with a half-life of almost 33 years. Is this also a real problem, or is this because 33 years is so long it's almost considered stable?
Okay, so I will first start with your first question. So there is the possibility to produce a generator to produce astatine. For that, you need to irradiate the bismuth target with a lithium beam, which means that you have to change the target from light charged particles to heavy ions. And this is something which is not very convenient. So there is only a very limited place where you can have access to the lithium beam at the moment. And then after that, you have some complicated chemistry in order to set up everything. So this is still a research area, and probably it will take some time before this can be made available. Okay, I know that there is people in Triumph that are working on that, but for sure at the moment what will be available is direct production of astatine-211, which will come, in few years from now. So we really have to focus on that if we really want to push astatine-211. Then for your second point is the bismuth 207. So that's true that could be an issue. So probably we will have to look at that country by country based on the regulation that you have in each country. So in many countries, people have started some clinical trials and they have not experienced any problem with that. But this is something that we should look more carefully and try to address in terms of dosimetry and impact.
Does it sound naive when I ask, can you filter it out maybe? They're always, let's say, long-lived radionuclides, something we have seen for the non-carrier, where there's a carrier added lutetium, right? We have this for some pathways to produce actinium-225. So far, there always have been certain solutions. Is there a solution to just make sure that we absorb it, the long-lived one?
Not to my knowledge, but I'm not a chemist, so that's something where we should. But the half-life is so long as compared to astatine that you have a very limited amount that is present in a patient. So really we have to look on the dosimetry to see what will be the impact of that, but it will be very, very low.
Tadashi, I also have a difficult question for you. When I read the available literature, it says when you think about the future role of astatine, it's more likely not in a, let's say, Me Too application of already established indications, like, for example, you mentioned thyroid or, for example, prostate cancer, but rather using this unique opportunity to actually go into rather orphan indications. And one of the reasons is you need quite some significant infrastructure investment to have this network of cyclotrons, and you have probably also still a comparably limited number of doses per year. And then this should be super successful for, let's say, rather orphan indications. What is your view there?
Yes. So in Osaka University, we started from using astatine for simple application as to use as an iodine analog for thyroid cancer. So it's a starting point. And next, I selected to you target PSMA. So the reason is we need to prove that astatine can be used, is effective other alpha emitters. So that's why I selected PSMA. But for the next target, yeah, as you pointed out, orphan drug or some targeting rare disease can be one of the options to get quick approval for our astatine label drugs. Yeah, that's one of the options. But uh, but I prefer the pan-tumor targeting rather than the orphan drug targeting. So yeah, but yeah, you are right. That orphan drug is one of the way to go.
Thank you, Tadashi. I also have a difficult question for Professor Sandmaier. So we mentioned excellent data for, I think, both the radiomonotherapies, yttrium-90, as well as the one using iodine. Then actually quite compelling data for the IOMAP, what the brand name is, the iodine 131, I think CD45. So one of the big problems always was the buy-in of the oncologists outside of UW. So how do we get the other clinicians, hematologists, really on board? What do you recommend us?
I think the hematologists, oncologists would be on board if it was something that was a little bit more simpler than the perception of having your patients in lead-lined rooms and they have to be isolated. And a lot of these patients are medically fragile. So if you have someone, because, we're doing transplants in patients now in their mid and even upper 70s. So they do have other problems. So if you can't go in, if nurses are afraid to go in, there's a lot of reluctance. But with astatine, the physics of it is it's not going to expose a person who's taking care of them in the same way as iodine. Doesn't require lead-lined rooms, and plus it's out of the system much quicker, And so I don't think there'll be reluctance of my colleagues to use it. In fact, I have people ask me all the time, why can't, can we bring that to our center? This is interesting. We'd like to do this. So I don't think you need convincing of us. It would be whether the colleagues at the other institutes who will help produce it, radio-label the antibodies. And to me, I really see the only issue is the production at other sites or the ability to produce at one site and ship to another. And my simplistic brain thinks that means making enough that when you ship it, there's enough half-life left in order to do the radio labeling.
So we always come back to the point of supply, right? I think the half-life of 7.2 hours shouldn't be such a huge problem, right? We have figured out the supply chain for technetium, 6 hours half-life. So I think whoever has done this can also do this for
Exactly.
Circle. I'm more concerned, and this is again, Dr. Haddad. So if
we rely on you.
we need investment, right? And I'm not saying we rely on your investment for Alnax. You're doing scientific pioneering work. But if I understand correctly, I looked at it as like they say you need 5 of these dedicated cyclotrons in the US to cover the US. You probably need 4 to 5 in Europe. If you want to go somewhere to Australia and maybe also Japan, you need 2 more. Each of them are around 20 million. So you need someone for a dedicated product to invest $240, $250 million upfront. How can we do this? Or from a European perspective, is this something where we should try to, we have the EU beating cancer plan and all this stuff. Is this something where we should try to pioneer and say we just we invest in infrastructure? It takes 2 to 3 years anyway to build it. And in the 3 years, we have enough drugs being ready to be deployed? Or what is your suggestion? You have so much experience in this field.
In fact, the point is that you have to build a business case around that. And that's true that you will have to invest, but you will also, if your drug is successful, you will have revenue that will be much higher than the investment that you have. So it's really a matter of business case. It's also a matter of having this set up gradually. So, some companies are just saying that they are building facilities, or you have this alpha-nuclide in China, you have this IBF Framatome in Europe, in the US, Ionetics, Nusano, and so on, and Sumitomo in Osaka. So they already have the first level of facility that will help to start with phase 1 clinical trial. And based on the results, then there will be, if they are successful, then you have more incentive to build new ones and so on. And then gradually you will reach to your network being installed and working. And in fact, if you look, for example, in Europe, we already have a network of 4 different facilities that are available. Few others can come also online. In the US, the same with the DOE program. There is already 4 to 5 facilities, and the same in Japan. So in fact, there is already some starting network that is there. They are not fully devoted to Astatine 211, but at least they can start the process. And then based on that, you can add gradually new machines for that are really devoted to Astatine 211. And that will make, in 5 years from now, probably Astatine widely available all around the world.
So the reason for my question was, it's probably difficult to decide which of the Astatine drugs is going to make it, but there are 10 or 15 right now currently developed, so one will make it. So, or let's say the bet is not as bold when you say this one is making it, if you say one of the 15 is making it. But each of them will actually need the network afterwards. So technically, it might even make sense to already build a network, betting that one of the drugs eventually makes it. And once they're ready, it's not easy to build a network within 1 or 2 years, as you know, much better than me. And then it would be good to have one.
Yeah, but if I can comment, I mean, if you look, I mean, there is already announcement by several of the companies that I have listed that say that they will make networks. So in fact, you will have 3 to 4 networks that are starting at the moment. So in fact, you will have that network in 5 years from now, probably.
Okay. So, I mean, the idea of this podcast today was really to make the field of nuclear medicine a bit more familiar with Astatine. I'm very grateful for the 3 of you contributing to this. I think we can already schedule a follow-up call in a year from now, because I think this is a topic which is coming back, and we will make progress. Nevertheless, thank you very, very much for being available. I want to highlight again also, self-referring from the Journal of Nuclear Medicine, the recent article by Richard Zimmerman, is 211 Astatine really happening? And nevertheless, it's super important to have 3 pioneers like you pushing this forward. So thank you very, very much for doing this. Thank you.
Thank you for the invitation.
Thank you very much. And there will be in November a World Astatine Community meeting in Nantes. So if people want to be
we will link it in the podcast.
Yeah, that will be fine because
absolutely.
That's the best way to learn about Astatine and to see all the community. That would be very important. Thank you very much.
And also Nantes is nearby Aronnax, right? So you will
also
have an opportunity to visit Aronnax.
Yeah.
No,
yes.
Perfect. We put this also in the subnotes or whatever of the podcast. No, but it's good. Absolutely.
Well, it's good. You have to inform the community or people that are interested in learning a bit more.
So thank you very much.
Thanks for watching, and thanks to IBA for sponsoring this episode. Please remember to like and subscribe.